Hello, welcome to Study Hub Pro. Today, we're diving into one of the most dynamic and frankly surprising parts of genetics, transposons. You know, for a long time, we pictured the genome as this static, stable blueprint, but that could not be further from the truth. It turns out our DNA is home to these restless little segments that can literally just pick up and move. I mean, it sounds like something out of science fiction, right? But this is a fundamental reality of molecular biology. It's happening inside our cells right now, and it has these huge implications for everything from evolution to disease. So let's get a proper introduction to these genetic acrobats. They're technically known as mobile genetic elements, but pretty much everyone calls them by their much cooler nickname, jumping genes. Okay, so what is a transposon? Well, it's a sequence of DNA that can change its position within a genome. Think of it like a small, self-contained genetic package. It comes with its own little molecular toolkit to essentially cut itself out and paste itself somewhere new. And they can range in size from just a few hundred base pairs to several thousand. What's amazing is that these were discovered way back in the 1940s, even before we knew about the DNA double helix. A scientist named Barbara McClintock was studying maize, corn, and she noticed these weird color patterns on the kernels that just didn't follow the normal rules of inheritance. She figured out that genetic elements had to be physically moving around, turning genes on and off, and those speckled kernels were the proof. And these aren't some rare genetic oddity, not at all. Get this, somewhere between 50 and 65% of the entire human genome is made up of these things. For years, scientists just dismissed it all as junk DNA, but now we know it's actually a massive engine of evolution, providing the raw material for genetic change. So, how do they do it? What kind of machinery lets these genes jump around? Let's take a look at their basic structure. Okay, the simplest version is called an insertion sequence, or an IS element. It's got two really key parts. Right in the middle, there's a gene that makes an enzyme called transposase. That's the molecular scissors and glue that does all the work. And on either side, you have these things called inverted repeats. You can think of those like little signposts that tell the transposase enzyme exactly where to cut. Now, it's really important to distinguish transposons from other bits of mobile DNA. Unlike plasmids, which are these little circles of DNA that can replicate all on their own, transposons can't. And unlike prophages, which are basically viruses hiding in the genome, transposons don't have a viral life cycle. They are purely passengers inside the host's DNA. So how do they actually move? Well, there are a few different ways. But today we're going to focus on two main strategies that are really common, especially in bacteria. Here's the general idea of how they insert themselves. First, that transposase enzyme we talked about makes these staggered cuts in the target DNA, creating little overhangs. We call them sticky ends. The transposon just slots itself right into that gap. Then the cell's own DNA repair machinery comes along and fills in the gaps. And a really key result of this whole process is that the little sequence at the insertion site gets duplicated. So you end up with these flanking direct repeats on either side of the transposon. Now let's look at a more complex type. Composite transposons are really interesting because they often carry extra passenger genes with them, things like genes for antibiotic resistance, and they're sandwiched between two of those simple IS elements. The whole unit moves together in what we call a non-replicative or cut-and-paste mechanism. It literally cuts itself out of one spot and pastes itself into a new one. The other major strategy is called replicative transposition, and instead of cut and paste, this one is copy and paste. The original transposon stays exactly where it is, but a copy is made, and that new copy is what inserts itself somewhere else in the genome. So with this method, the number of transposons actually increases over time. So what happens when all these genes start jumping around? Is it a good thing? A bad thing? Well, as with a lot of things in biology, it's kind of both. Let's look at the consequences. A single jump can have some pretty dramatic effects. If a transposon happens to land right in the middle of a working gene, like the APC gene shown here, which helps prevent tumors, it can completely break it. This insertion creates a mutation, basically turning the gene off, and that can lead to diseases like cancer. So to sum it up, their impact is huge. They're a major source of mutation and genetic variation. They can cause big rearrangements and deletions of DNA. And, really critically in medicine today, they are a huge reason for the rapid spread of antibiotic resistance genes among bacteria, because they can jump between the main chromosome and those plasmids we mentioned earlier. But
Here's the cool part. Scientists have figured out how to take this natural mechanism and turn it into an incredibly powerful tool for research and even medicine.
We can actually use transposons to intentionally break genes just to figure out what they do. That's a process called insertional mutagenesis. They're also used for gene mapping, cloning, and even transferring genes between different species. They've become this absolutely essential part of the modern geneticist's toolkit.
And this shows their incredible potential for the future. In gene therapy, we can actually re-engineer a transposon system. We can take out its normal jumping genes and replace them with a healthy copy of a gene that a patient might be missing. The transposon then acts like a high-precision delivery vehicle, cutting and pasting this gene of interest right into the patient's cells. It's a really promising way to permanently fix the root cause of many genetic diseases.
So, from causing speckled corn to spreading antibiotic resistance and now offering hope for gene therapy, these jumping genes have a really complex legacy. They were the drivers of evolution for billions of years, creating change and diversity. The question for us now is, how will we use what we know about them to shape what comes next? See you in the next video.